Hello, everyone. Welcome back. As people file in here for the second part of our session, I just want to say I hope you all had enough time to enjoy all the activities. If you are a neuroposter judge, if you haven't turned in your scorecards, could you please raise your hand now? And one of our volunteers will come around to collect them. So if you have scorecards for neuro judges, raise your hand. Okay. So with that, I'm really excited to introduce our next speaker, Dr. Lin Tian. Dr. Tian is a professor of biochemistry and molecular medicine and an affiliated faculty of the Center for Neuroscience. Dr. Tian's research focuses on engineering optical probes for monitoring and controlling neural circuitry in living, behaving animals. These new imaging techniques have greatly impacted the field of neuroscience, facilitating new types of biological experiments performed to address previously intractable questions. Today, Dr. Tian will share how her research is helping to reveal the mechanisms underlying behaviors and serves as a drug discovery platform for identifying new therapeutic targets. So with that, I will give the stage to Dr. Tian.
Thank you. Thanks, Tim, for a very nice introduction. I would like to thank all the committee members for giving me this great opportunity. Can you hear me now?
Is it on?
I think it's on. It's on. Let's see. Can you hear me now? Better? Oh, I can use that, so maybe it's better. Hello? Okay, good. Yeah. All right, much better. Yes, so I would really want to thank all the committee members and all the graduate students and give me this great opportunity to share what light sensors engineered with machine learning and teaching us about the mind and how the current biotechnology really shaping the future of neuroscience. Sir. So, from the previous talk, you have learned about connectome, right? So this is a macro scale of connectome. And from here, we really learned, how we can use the color and the light to build this roadmap to figure out the general architecture of the brain, the circuit, which we call connectome. But brain is very complex. We also learned that, right? So they're inaccessible and also very fragile. So the study of the connectome is a great example to show us that how neuroscience and optics can come together to overcome these challenges, to tell us or reveal the richness and diversity of the brain anatomy and also the structure. However, having this static map is only a small piece of this puzzle, right? So it's just a static map. So in order to understand the function of the brain, so for example, how we can just move our leg and move our finger, we must catch a dynamic picture of the brain in function, right? Actually, it will be better, we can use light and color again to penetrate into the brain to watch how the neurons actually communicate with each other in real time. So how do we do that? So before I tell you about the sensor story, I want to let you know what you see here is called the synapse, right? So this is a functional unit. It's also a structured unit of the brain where two neurons connect to each other. We call synapse. But there's a lot more about this structured functional unit because this is the way how neurons communicate with each other by releasing chemical transmitters, right? So you probably already heard those chemical transmitters such as dopamine, serotonin, and glutamate. So those neurochemicals actually conserved across species. The dopamine in a fish, in a monkey, is the same as the dopamine in human. Why is that? because those neurochemicals are very important for some essential behaviors such as foraging and survival, and they are the one to help your brain to quickly adapt, to a new state when something just occurred, and to help you to realize what's actually going on around us. Because you see those connectome, this is a wiring diagram of the brain. But in order to make us who we are, what's really working is those neurochemicals. They release in time and space to quickly adapt the brain to the state so we can know what's going on around us. So they're so important, we can think about what if those signal goes wrong? So those neurochemicals are actually implicated in literally every single neuropsychiatric disorders. Right? For example, depression, Parkinson's, schizophrenia, or Alzheimer's disease. All of those diseases is actually related to dysfunction of those neurochemicals. So for the rest of my talk, I'm going to use serotonin, a serotonergic system as an example to tell you how we can develop technology to be able to see the action of those neurochemicals and how we can reveal how the drug actually changes the brain. So what I'm showing you here is a 3D rendering of a serotonin system, in the left hemisphere of the mouse brain. I'm going to play the movie. So you can see what we showed here is all the serotonin projections, those axons, go from a very small structure called dorsal root in the brainstem, like very deep here, but it's project almost every single brain regions. Every single brain region have this serotonergic projection from a very tiny, teeny brain structure called dorsal root. And if you take a close look, they will communicate with local neurons, again, through the synapse I just told you. But this actually is the target of all those broadly utilized treatment for depression and anxiety. For example, what I showed you here, you can see this is fluoxetine. So fluoxetine is one kind of serotonin reuptake inhibitor we call SSRI. So this is, in the past 40 years, this is the major drug that's actually been broadly utilized to treat depression. However, you may also know those drugs they only work partially effective, right? So first of all, maybe only 30% of patients will respond to the treatment of SSRI, such as fluoxetine. Even with 30% that responded to the treatment, the action actually takes a very long time. So the effect does not kick in probably about 3 or 4 months later, right? So it's very slow. The patient have suffered for 3 to 4 months before they will know, okay, the drug going to work or not. And recently, there's another type of drug called psychedelic. So those drugs also target to those serotonergic synapses, same thing as SSRI. But different from SSRI, those drugs is fast-acting, which means once you have one dose of those drugs, you will feel so happy and so content, you will not depressed anymore. But the problem is those drugs also give you hallucination, which means you will hear or see something that you think is so real, but it actually does not even exist, right? So those are the side effects. So what the neuroscience is facing these days is not only we need to develop fast-acting therapeutics, right, to treat depression, but also we need to avoid unnecessary side effects such as hallucination. So how do we do that? So in order to develop effective therapeutics, we first have to really understand how the serotonin works, right? We need to have a very better understanding of the serotonergic circuitry and how the serotonin is released in time and space, in the living brain. And second, we also want to know how those drugs, such as SSRI, really modulate or change the release pattern of serotonin, right? And then we also need to develop a better assay to really avoid this hallucinogenic effect. because you cannot wait until you give the drug to patient, to human, and ask, do you have hallucinogenic effect, right? You want to be able to do this at the early stage of drug discovery to work with a chemical scaffold that may not lead to hallucination. So how do we do that? So my lab is actually trying to fill those gaps in knowledge by developing the light-based technology. So we built those sensors, those protein-based sensors, so we can, with a microscope, we can see when and where the serotonin is released. So how do we do that? Again, we start to borrow from nature. So we borrow those fluorescent protein from jellyfish, we call the green fluorescent protein. And also we borrow a serotonin binding protein from E. coli, which is used as a ligand binding. And then we play tricks called protein engineering, and then we can build a sensor, so which is this, we call this light sensor, which is actually a protein that can provide an optical readout of extracellular concentration of serotonin, which means now if we put this in your brain with a microscope, we can see exactly when and where the serotonin is released. If you feel happy now, that means the serotonin is actually at work. So, but what's this to do with machine learning? Because we use machine learning to really be able to speed up or accelerate our work to be able to build those sensors. Because you can see we borrow two different components from nature, we put them together, but we will not have the power to predict how the sequence, which is amino acid sequence, how that can reflect to a 3D structured protein or predict their function. So we will be able to use those machine learning to be able to accelerate and scale up the process from design a protein on a piece of paper to go through the wet lab work and finally make a protein that we think how it works. So because of this artificial intelligence, we can actually feed the computer tons of data about the relationship of a sequence and the function, and we use this machine learning to predict where we should manipulate or alter and to design this protein that's actually as the way we hope that can perform. So this technology allows us to be able to speed up this protein engineering process. In the past, the field takes about 10 years to build those sensors, but now we pretty much can build a prototype in 6 months. So you can see how the technology can really lead us to really speed up this development. And then we next will validate the sensor. So we put the sensor into a model animal. Here we show is a mouse. So we put the sensor into the mouse brain. And after that, we put a microscope on top of it. And the mouse will get, freely behaving, which means the mouse will be able to do anything it wishes. But then when we do it, we want to monitor how the serotonin fluctuates when the animal goes through different sleep and wakefulness cycles. So what you see here is actually the serotonin fluctuation from awake to sleep, right? The different stage. So then I want you to focus on the right side. So you can see that serotonin level increase so much within 10 seconds when you start to go from the NREM to the wakefulness, right? So as soon as you wake up, your serotonin level actually can significantly goes up within seconds. And when you go to sleep, your serotonin level can significantly goes down within another 10 seconds. So with those sensors, we really can push the sensitivity and also resolution, to sub-milliseconds and a second to see how those serotonin is actually released. And then we want to put those sensors to the use. And one of the questions we're trying to ask is, how do we know which SSRI we should subscribe to the patient? Because we all have different genetic background, which means the one essential protein called the SERT, which is the transporter, we're thinking that's where SSRI actually target. the SERC protein in my brain is probably different from the SERC protein in your brain because we have different genetic makeup. So what we do is we can actually take the sensor and combine it with the human iPSC cells, which means we take the cell from your skin sample and we derive into neurons, so make a mini brain in the dish. And then we put a sensor into this neuron. And now we can create this serotonin isolation. And then we can put the drug or combination of the drugs to those cells, and we can monitor whether they're active. So you can see this SSRI actually can really block the serotonin transport, right? That means the serotonin will be able to accumulate at the synapse, and that can give some modulation effect to make you feel happy. So this is really powerful because think about those treatments sometimes does not kick in until like 3 or 4 months later. And you don't know, the doctors do not know which SSR will actually work. So we cannot take a small sample from skin biopsy, we grow those cells in a couple of weeks, and we can test 100 of those SRI, and we can test them in different dose, we tested them in different combination, and now we can see, oh, this one actually works better for you, before they subscribe those drugs to you. So you know they will work. So you have the hope those drugs can work. Okay, so next I want to take you to travel to psychedelics. So another type of drug that's actually targeted to the serotonergic synapse is called psychedelic. One of the problems with psychedelic is hallucination. They're a fast-acting antidepressant, but also cause hallucination. As I said, you can hear or see something that does not exist. So the question is, how do we test those things, hallucination, in a test tube, right? So we can predict hallucination potentials of any kind of drug or chemical scaffold before we give to animal or give to human. So again, we build a sensor called Cycli. So again, we borrow from nature, those fluorescent proteins. But this time, instead of borrowing a serotonin binding protein from E. coli, we borrow a protein called serotonin 2A receptor from human. Right? So this serotonin 2A receptor is a major target of a psychedelic. The psychedelic will bind those receptor and induce some signaling cascades that will lead to both antidepressant and also hallucination effect. And from there, we build a sensor called Cycli. So you can see when the serotonin or psychedelic bind, it will create an optical readout of the sensor. So we know because this optical readout is different from drug to drug, we can set up a high throughput screening assay, and we can really predict the hallucinogenic potentials of this drug just in a test tube. So the underlying mechanism we're still trying to explore, but it tells us how the drug bound to the receptor. Already, this conformational change can actually dictate to the ultimate brain, behavior output. So from there, we can have drugs as hallucinogen, or the drugs predict as non-hallucinogen, and combined with all kinds of different animal behavior, we can finally confirm whether the drug is hallucinogen or it's not a hallucinogen, but also remains antidepressant effect. So ideal drug, for example, SSRI, it's not hallucinogen. It works, antidepressant, anti-anxiety, it works, but it's very slow, right? Ketamine, I think, many people, if you had a ketamine, for your treatment or surgery process, you probably have a very wild ride during, that time. So ketamine, your hallucination increase. It's a fast antidepressant. But what's the ideal drug? If we use those assays to screen the ideal drug, then we can confirm, they work really quickly, but they may not have the hallucinogen. They are not hallucinogen, so they don't induce hallucinogenic effect. Again, very effective therapeutics, without unwanted side effect. So that's what we wish that's going to happen in the near future, fast-acting antidepressant. But together, we need those technologies. So this shows some startup company funded by graduate student and can already start to work with this Lundbeck is a big pharmaceutical company in Europe. So this is a very good example to show you how the UC Davis can position the Center for Neuroscience at the interface of startup and collaboration with industry. So moving into the future, I really envision colorful and also brighter investigation or analysis of those neurochemical released at the synapse. So think about with the color, right? So the color is a spectrum. So now we actually can start to create those sensors that actually have different color. So we may be able to look at 5 or 10 or even 100 of those released neurochemicals at the same time when the rodent model is performing behavior. So from there, we can really start to detangle a very complex neural circuit. And just with the sensor, again, this machine learning, right, will provide tons of those data sets. Those data set is really valuable for theoretic neuroscientists, where you're gonna hear from Rishi's talk. There's a basis to be able to develop a new hypothesis or refine the theory about the brain computation, right? So you can see all those things, those sectors needed to work together to be able to integrate in order to tackle those big questions about how the brain works and how we can build the effective therapeutics. So without that, I would like to thank my lab and also the funding. This is really, all the work I told you is actually done by the graduate student and postdocs in the lab. And they are the one actually make all those things happen. Those young minds, the trainees actually made everything possible. And specifically, I want to point you to all those faces you may already met at the NeuroFest. With that, thank you for your attention. I can take some questions.
So again, raise hands if you have questions, and we can bring a mic to you. I see one here.
What does SSRI stand for?
It's called a serotonin reuptake inhibitor. So basically, that's the type of drug, a class of drug that's actually bound. We think they bound to those serotonin transporters. So when they bound those transporter, right, the serotonin cannot be reuptake. So serotonin released at synapse to outside of neurons, and they have to come back to the neurons through those transporters. So now if you block those transporters, serotonin concentration will increase. Then we think that's how it works. Once it's increased, you feel happy. But we do not understand why it takes such a long time to kick in and why only 30% patient is effective. So which means there's other mechanisms, maybe the major mechanism, we do not understand how those drug works. But the brain chemistry, all those, therapeutics, it's the same in the past 40 years, right? Everyone using SSRI to treat depression. So we're hoping those new technology will finally, bring us to a new era of drug discovery in terms of treatment. Because now we can see with such a, high resolution when and where those chemical is released and what's their function. Yeah, and how they react to the drug. Yes?
What technology might be used now to find the chemicals that we don't yet know about? Are there light sensors looking for things that work in tandem with serotonin?
A really good question. So that's why we need all those different color spectrum, right? Because at the synapse, it's not just serotonin in action. There's glutamate, there's dopamine. They can be released at the same time. You know, how they collectively modulate the brain states, we have no idea. So that's why we need the machine learning to help us to build more sensors and to be creative to have a palette of color. And then now it's light, right? It's a light and color. So I feel like this is just amazing how the optics and neuroscience come together to make the brain accessible. Yeah, so that's the future work we're hoping to do is colorful brain, and each color will be one transmitter.
So you've used together the GFP with the like the neurotransmitter protein. How do you know that there's no like steric hindrance from the GFP when it's activated, or that the newly spliced protein still acts exactly the same way as the wild type?
Yeah, that's actually a good question. So that's why we need to make thousands of those proteins, because we don't, as you said, we do not know which one work. Everything coming from a sequence, right? You're trying to design something, you hope that will work. The ligand binding will lead to changes of fluorescence. So we do a lot of high super screening to look at multiple property of the sensors. And then we move on, we kind of take something, we screening them, we look at all different function, property, and then we're moving one winner forward to the brain, right? So that's why machine learning can really help us. It's predictive power from those machine learning methods.
You mentioned how the drugs such as ketamine or SSR, they trigger serotonin release in the brain. You also mentioned how the side effect of that is hallucination. I'm wondering, how exactly does the release of serotonin affect your sense of seeing or hearing?
So that's actually a good question. So basically, the sensor will see the release of the serotonin, right? When serotonin is released, the sensor will light up. If the serotonin get reuptake by circ, the fluorescence goes down, right? So it's kind of you think about like firefly, right? So what you see exactly under the microscope is a firefly. It's a flashing. So each flashing, that's one event means serotonin released. Then serotonin not released, serotonin released. So we can see when you have this SRI, you put in the dish, or you can give the animal, you will see how the serotonin oscillation can be changed, right? So for example, I showed you that with iPSC cells, what happens is if you put this SRI on the cell, which are human neurons, and your sensor all of a sudden does not light up anymore because there's no release, right? There's no transport going through the cells. But then you wash those drugs away, you see those flashing again. So it's a really kind of a light effect. Think about like firefly, blinking in the dark. Yeah, so brain is really dark. It's basically a big blob, dark tissues. Actually, those neurotransmitters, you cannot work with them on the bed, in the air, because they can be oxidized within 1 second. So only in the brain, because the brain is basically, that's why you have blood-brain barrier. It's everything is so insulated. It's a black, dark box. And you shine the light with the color, trying to reveal what's actually happening out there.
Yeah, we have time for one more question.
Great presentation. A quick question. I wonder if you could give an example of how machine learning plays a role in personalizing these quick action antidepressant drugs.
Yeah. So basically, this type of machine learning, again, I think Tim gave a really good talk today, explain what the machine learning means. So you basically have a lot of input data. You don't know the structure. You won't have the output, right? So same thing for protein engineering is you will have all those sequence. It's a 2D sequence, amino acid sequence. But how those sequences can, when you put them together, they can become tedious, like 3D structure of protein, and you can also predict the function of the protein, right? So we're trying to engineer a sensor. We want to engineer the sensor has certain property. For example, they're very fast, right? You want to, because serotonin releasing in seconds, you want to be very fast. You want the sensor to be very bright, so you can actually see with the light. You want a sensor to have a chemical specificity because they only light up to serotonin but not to dopamine, right? So basically we generate a lot of data first, and then we feed into the computer, and the computer predict, okay, those sequence in this will have this function. So classify different function. And we combine with AlphaFold, right? AlphaFold, again, it's actually machine learning, and we put sequencing, and we look at structure, and then it's kind of like iterative process. So usually we screen, so for the work I just showed, we screened about, I think 20,000 proteins to get that one sensor that works as we wanted. Yeah.
Thank you very much.